Oh, whoops. Okay. Hey, everybody. Sorry about that. Welcome back to part 2 of lecture 10. Now we're going to talk about neural pathways. So at the end of 10A, we were talking about the cells of the retina. So of course, the retina being that inner layer of the eyeball that has 5 layers of cells. And those photoreceptors are down here on the bottom. So rods and cones, and then they're going to send their signal to the bipolar cells. Hello. And then the bipolar cells will send a signal to the retinal ganglion cells, which then send an action potential, hi, along their axons, which becomes part of the optic nerve. I know. It's very exciting. So now we're going to talk about where do these signals go from here. So where do these axons travel? So you remember that the optic nerve is found on the bottom of the brain, cranial nerve number 2. And so I like this diagram here on the left because it's kind of simplified things and brought it out of the brain for you to see. So we've got 2 optic nerves, one from each eyeball, which are carrying axons that have action potentials traveling this way. Then there's like this X, this kind of crossing that we see, and that's called the optic chiasm. You can also see it over on this diagram here, that optic chiasm. And then the other kind of limbs of the X, as it were, that are leaving are called the optic tracts. So the order is optic nerve, then optic chiasm, then optic tracts. Now, in order to understand how these axons travel through these different structures, we have to talk a little bit about visual fields. When you are looking out at the world, you do not see like a square thing, like the image that you're seeing on your laptop. What you're seeing is actually kind of a circle, because you can see a certain amount this way, you can see a certain amount this way, which is what this diagram is showing you. And of course, the things you're looking directly at are the things that you see in very sharp detail, because that's where those light rays are hitting your fovea, where you have all those cones. But your field of vision is actually kind of circular. So a little bit earlier today, I took a couple of photographs of what my 2 eyeballs were seeing. So my 2 eyeballs are in slightly different positions, and so the perspective is slightly different. So one photo I took to represent what my left eye is seeing, and the other one is what my right eye is seeing. And so they're from slightly different perspectives. You may remember me discussing earlier when we talked about strabismus, that is how our brain is able to kind of calculate and estimate depth. So our depth perception is based off of some pretty sophisticated thinking that our brain does, comparing these 2 perspectives to each other to figure out how far away something is. Yes. Okay. So these are, basically the fields of vision of what I'm seeing in my left eye and my right eye. Now, to be honest, I took these in portrait mode with my phone, so it's not exactly, you're not going to have this fine detail that you can see, and you're not going to have as vibrant colors as you can see in these images. This is also, I kind of did these 2 ovals. It's not quite like that. So if you look at this diagram up here, your nose is in the way. So your right eye can only see kind of from here around, and your left eye can only see from here and around. So it's a little bit different, but you get the gist. So if I were looking at a world where in front of me, let's just say there's this huge billboard, and the left half of it is all blue and the right half of it is all red. Okay, so what I've got here is that what my left eyeball is seeing and what my right eyeball is seeing. So that's the visual fields for each individual eyeball. So we've got blue on the left, red on the right for each eyeball, because they're looking at the same thing. Okay, now when we look at how this ends up coming onto the retina, remember the lens flips everything upside down and backwards. So now we're just talking about right and left, so it's going to flip things backwards. We don't have to deal with the upside down part quite yet. So light from the left, the blue side of the billboard is hitting the right side of my retina. The right side of my retina. Now it gets a little confusing because what you'll see, and they talk about it in the book, is we talk about the temporal side, meaning the side of the retina closest to your temples, or the nasal side. So you can see that this blue on the right eye, this is my right eye over here, is hitting the temple side, where the one on the left is hitting the nasal side. Yes, so that gets a little weird. And then of course all the red light as it's coming in is hitting the other side of the retina because everything gets flipped upside down and backwards. Okay, so now that we understand where light from what I'm looking at is hitting my retina, so the red light, the red is hitting over here and the blue is hitting on the other sides, we're going to track where those axons from those different parts of the retina go. Okay, so this is a different diagram. I liked it a little bit better. They use orange and teal. So instead of a red and blue billboard, let's imagine you're looking at a world. The entire right side is this orange color, or peachish color, whatever you want to call it. The entire left side is teal. Shh, 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 shh. I don't know what he wants. Okay. So again, remember, so let me use blue, right? So the blue is coming in and hitting this side of the retina, coming in. It can't actually come in because it's too close. It would have to be farther away. And I'll just use red for the other one, right? It's hitting this side, and that would be hitting this side. Okay, so you can see I've kind of color-coded them in with the teal and the peach. So what I want you to pay most attention to now is where those axons are going. So let's start with the left eye over here, looking at where those kind of peach or orange axons, or axons carrying that information, are going. So they're back here on the back of the retina. They travel in through the optic nerve to the optic chiasm, stay on the left side, and go back into the left side of the brain. Now let's look at the ones on the other side. So here are the ones detecting kind of the orange color. Here they are on this side. And this time they come through the optic nerve here, but then they cross over and end up on the left side of the brain. Isn't that crazy? So let's trace the blue ones now, or the teal. So I've got my blue or teal on my left eye over here. The axons go through the optic nerve and they cross over to the right side. And then the ones from the right eyeball over here, they come through, but they stay on the right side. So what we see happening is that the temporal part of the retinas, those axons don't cross, but the ones that are on the nasal side cross, which is pretty crazy. So the end result is that everything that's on the right side of our visual field ends up talking to the left side of the brain, and everything that's on the left side of our visual field ends up going to the right side of our brain. So I will tell you, I think I caught that in one of the crash course videos, Hank Green actually says that the sensory information from the left eye goes to the right side of the brain and vice versa. That's not true. It splits. So it's actually the left field of vision goes to the right side of the brain, and the right field of vision goes to the left side of the brain. Crazy. All right, a little bit of vocabulary here. The word ipsilateral means on the same side as, whereas contralateral means on the opposite side of. So the visual field from my left ends up going to the right side of my brain, so that's contralateral. But if we're talking about some of those individual axons, right? So these axons on the temporal side of the retina end up staying on the ipsilateral side of the brain. Yeah. And same thing with the temporal side on the other eye, temporal, they end up staying on the ipsilateral side here as well. Kind of cool. So where do we go from there? So these are the optic tracts that we were just moving through. When we switched sides or didn't switch sides, we were moving through the optic tracts, which are not labeled on this particular diagram, but the optic tracts are here and the one on the other side. So you can see again that kind of red and blue and where each side goes. So from here, those optic tracts are going to go to the thalamus. So you might remember that when we first learned about the thalamus, which is part of that diencephalon, part of the middle part of the brain, that it serves to kind of filter out extraneous sensory information, and it kind of decides or triages which sensory information is going to come to the cerebral cortex so that we can be consciously aware of it. So this visual information is no different. So it's coming into the thalamus, specifically a structure called the lateral geniculate nucleus. So remember, one of the definitions of the word nucleus is a group of neurons in the brain that are all working together for a certain function. That's what we've got here. So here, those axons were the axons from the retinal ganglion cells that maybe crossed or maybe didn't. The next place they come is the LGN, the lateral geniculate nucleus of the thalamus, and then they're going to have their first synapse. So you're going to synapse on some neurons here in the LGN. All right, so then what? Okay, so from the LGN, which I like this diagram better. So here's the LGN here. So we've got one on each side. Then we're going to send out what are called axons and what are called optic radiations. So radiations, because they're kind of like radiating out, and they're not necessarily all bundled together, although they kind of do move through two similar kind of bundles. So you can see we've got kind of axon here and an axon here, and they are sending these signals back to the occipital lobe. Because remember, when we talked about lobes of the brain, the occipital lobe is the one for processing vision. Specifically, it's going to go to what's called the primary visual cortex, which is also referred to as V1. So from the LGN in the thalamus, axons then send signals back to the occipital cortex, to the primary visual cortex, or V1, and this is where perception begins. This is where you first become consciously aware of, oh, I'm seeing something. So that's going to take us into perception, which is going to be the next and final part of the lecture. So this is our review slide because it was too hard to kind of write out. So we're imagining that the entire right side of our visual field is blue and the entire left side is red in this particular example. So the red is hitting this side of this retina and this side of this retina. So when it's the temporal retina, which is the second one, it's going to stay on that side and go back through the optic nerve, optic chiasm, to the lateral geniculate nucleus of the thalamus, through optic radiations, and then to the primary visual cortex. The red that we're seeing with the other eyeball is going to go through the optic nerve, is going to cross at the optic chiasm, go to the LGN of the thalamus, and then send radiations back to the primary visual cortex, otherwise known as V1. So again, let's look at blue. So the blue over here on the temporal side is going to go through the optic nerve. It's going to stay on its ipsilateral side at the optic chiasm. It doesn't cross. Goes to the LGN of the thalamus. Optic radiations back to the occipital lobe to V1. And then the one over here, which is the nasal side of the retina, is going to go through the optic nerve. It crosses at the optic chiasm, goes through the optic tract to the LGN of the thalamus, which then sends out a radiation, which goes back to V1, or the primary visual cortex in the occipital lobe. So those are the neural pathways. Crazy. I'll see you next for perception.